quality control and assurance. So QC is associated with internal activities that ensure diagnostic test accuracy, and quality assurance is associated with external activities that ensure positive patient outcomes. And based on this, there is a different things we can notice, that is reduced length of the patient stay, reduced cost of stay, reduced turnaround time for diagnosis of infection. It plays a very important role, and to appropriate antimicrobial therapy for the patient. Finally, there should be the customer satisfaction, whether it may be physician or patient. Sorry, screen is freezed. Please wait a bit. So now go for diagnostic cycle. And diagnostic cycle is based on three phases, that is pre-analytical phase, analytical phase, and post-analytical phase. So we will see the each and every phase step by step. So if we come for pre-analytical phase, it comes different parts like specimen collection, that is collection of the sample, transport and the verifying the details, observation of the sample, and ruling out the criteria for rejection. And first, the specimen collection. So specimen collection is very important, and among that, site of collection is very important. And in microbiology, collection of the correct sample is very important, and minimum contamination we have to see collect the sample. So in case if you want to send the sample for pus culture, preferably aspirates are preferred than pus swabs. And in the collection of urine culture and all, a proper guidance has given to the patient and to collect stream urine sample, clean cash urine sample. And next, time of collection. Time of collection also plays a very important role, and the collection of the time should be optimal. For example, if we suspect the patient suffering with typhoid, then in the first week of the fever, the blood cultures will be positive. In the second week of the fever, the serology will come positive. In third week, stool cultures will be positive, and in fourth week, urine culture will be positive. So based on this, time of the collection is also very important. And quantity of the specimen, it also plays a very important role. So proper guidelines has to be maintained to collect the sample or to send the proper appropriate sample. If it is low, then it may not be sufficient to the processing for various investigations. And if at all we are sending different culture samples, we have to check that the sample should be collected for the antibiotic administration. And last, the proper labeling. It also plays a very important role. So for labeling, most and important thing is identification number should be correct. Name, age, gender, nature of the specimen, date and hour collected, diagnosis has to be written, and if at all any antibiotics has given, and also any investigation required should be labeled on the container. And next, the transport of the specimen. So specimen transport is very important here. Why? Because it also plays a very important role. Actually, we have to send the sample to the laboratory in its original state, and adverse environmental conditions such as extremes of heat and cold, and if at all it is transporting through the air transport, rapid changes in pressure and excessive drying should be avoided. And for prolonged delay, and if it is more than 4 days, it should be minus 70 degrees centigrade. Next, the criteria of sample rejection. So sample, based on the collection of the sample, there is some criteria to take the sample or to accept the sample. If at all Any cultures received in formalin should be rejected. Similarly, 24-hour sputum collection and smears of secretions from uterine cervix or vaginal or anus for gram stain and for the detection of Neisseria gonorrhoeae. And a single swab is sent for multiple investigations like for aerobic culture, anaerobic culture, fungal culture, and for mycobacterium tuberculosis that is not accepted. And in some cases, like improper collection site, like stool collected from different site, like respiratory syncytial virus, means nasopharyngeal swab is sent instead of stool. So similar like this, there are different ways where the samples can be rejected. And there are also some other ways, like container is leaked or unlabeled or wrongly labeled or mismatched. And similarly, quantity not sufficient. And if there is any prolonged transport, and if it is drying of the specimen or the hemolyzed blood or lipemic sample or wrong tube. So we have finished the pre-analytical phase. And if we comes for analytical phase, after receiving the sample to the laboratory, and especially in microbiology, first thing we have to check for the microscopy. So based on that only, we can also see whether it is yeast or positive organism or gram-negative organism, like that. And next we have to process the samples based on the using appropriate culture media. And next, we'll do the preliminary identification by doing various testing. And after incubation of the culture via plates, we will interpretation the cultures by seeing the colony morphology of different organisms. And based on that, we will do the antibiotic sensitivity testing. So these all come under the analytical phase, especially for the cultures in microbiology. And as today's topic is quality control, so I'm just concentrating on the QC of various techniques, like QC of stains. So staining technique is very important in microbiology. So here a few things I want to discuss with you. All stains and reagents must be clearly labeled, dated, and stored correctly. And it should not be used beyond their expiry date. And it should not be used when they show signs of deterioration, like abnormal turbidity or decoloration. And at regular intervals and whenever a new stain is prepared, control smear should be stained. And smear should not be too thick. And especially in the gram staining, decolorization plays a very important role. There, if it is incomplete, the gram-negative organisms may be reported as gram-positive. And to maintain the quality control of the stains, as per the CLSI guidelines, and we are using some standard strains like ATCC strains for getting the expected result. Example, I will tell you about gram stain. So for identifying the gram positive and gram negative, or use the ATCC strains for Staph aureus strains for gram positive and E. coli strain for gram negative. So that we have to cross check, means we have to prepare the strains, smears with these strains, and we have to do gram stain, and we have to get expected result. Similarly to zeal stain or whatever other stains we have to do, we should use ATCC stains, and we have to do the proper staining technique, and we have to observe the expected result. And these are all the biochemical tests which we commonly do in the laboratory. So even for biochemical tests, so if we see catalase, coagulase, indole, these are all biochemical reactions which undergo in microbiology laboratory. So even for this, we will maintain the standard or we will maintain the quality control by using the test control organism. So for each and individual investigation, a different strain is used, and we have to check for the expected reaction. So these are all the results, expected reactions, and the test control organism used for various biochemical reaction. And next comes the quality control of equipments. Equipments also plays a very important role because to get the proper result, especially in microbiology, incubation and all plays a very important role for the growth of the bacteria. And if it comes, the quality control surveillance procedures for microbiology equipment, we have to check for the quality control. There will be a schedule. Either it may be daily schedule, it may be weekly schedule, or monthly schedule, or quarterly schedule will also be there. For example, I will tell refrigerators. So recording of temperature, it is a daily basis. So we have to place a chart on the refrigerator, and daily we have to check it morning and evening, and we have to note down temperature. Similarly, incubators, it is also daily. And if we come for autoclave, at least weekly we have to check it by using the test strip with spore strips. So, and we have to check that the no growth of spores will grow. And similarly, monthly, like centrifuges, there will be a tachometer for checking the revolutions, and we have to see whether the RPM is getting correct or not. Means it should be within the limits. And first of all, I want to tell you one more thing. These are all the quality control we are checking internally, either daily, weekly, or monthly. But annually, we have to get all the equipments done, calibrated, either from the outsiders, so that we will get a proper idea whether the quality of the equipment is correct or not. I hope you understood that this is the quality of control of equipments which are done schedule wise based on daily, weekly, and monthly. And next come QC of culture media. Culture media plays a very important role in microbiology. Why? Because we are providing the food for the bacteria to grow. So the proper precautions we have to take to grow the bacteria. Especially water used for the media should be free of ions, and the pH of the water should be slightly acidic side, and it should not be less than 5.5. And media prepared should be free from excessive bubbles or pits. And we have to see that it should be free of cracks, freezing, or crystallization. And while pouring the media, we have to see that the thickness of the medium should be 4 mm, and especially we should prefer the borosilicate glassware for the preparation of the media. And similarly, after preparation of the media, we have to check the quality of the media and to check the characteristics of the organism in the particular media. Here also we will use the standard, that is 80cc strains, different strains for different media. Like there are different media we use, like blood agar, chocolate agar, Mekong agar, and various other agars are used. And to show the media prepared, whether it is showing whether it is giving out a complete characteristic appearance of the organism. Here also we are using the 80cc strains. First we have to inoculate the strains, and we have to check whether the media prepared by us is correct or not. Okay. So that is all I have just discussed about QC of stains, equipments, and culture media. And there is a lot to discuss, but as we are less time, now I want to go to post-analytical session. So after pre-analytical, post-analytical, I just want, analytical, I want to go about post-analytical. So finally, after analysis, reporting of the results has to be done. And next, interaction with the clinician and regarding diagnosis. So after interaction with the clinician, we have to analyze the and we have to go for the sample storage. And after sample storage, we have to maintain the record. So here also we will see the step by step. So reporting of the results play a very important role. Actually, I just forgot to share a screen. Here in our hospital system, we are using the Dhanvantri. So it also helps us a lot to maintain the turnaround time. Why? Because giving manually and all, it takes a lot of time. So our hospital information system that is based on Dhanvantri, we are issuing the reports online so that the patient get the reports very quickly. And even for our equipments, we have LIS system, so that directly it will go into the Dhanvantri and the patients can collect the report very easily. So it plays a very important role. So reporting of the results, it should be quick, and the terminology should be understandable. It may be, reports may be designated as a urgent report. So emergency based we have process and we have to give the reports. And urgent reports must always be telephone, means especially for the accreditation NABH critical care registers we are maintaining. So if in case if you think that it is a very critical or emergency situation to inform the results, we have to telephone and inform it to the wards. And confidentiality of the patient data should be ensured, and timely preliminary interim report should be issued, and especially reference range should be given wherever it is appropriate. And a copy of report should be dispatched or should be kept in the lab. And maintenance of samples and records. So after result giving the reports, so record should be maintained. There will be a guidelines, that is either national or local guidelines must be followed, and as per the hospital, record should be maintained for at least 2 years, ideally for 10 years. Now, according to accreditation, it depends upon the hospital to hospital policy. It may be 3 years, it may be 5 years, but at least 2 years, and ideally for 10 years, all the patient records has to be maintained. And when comes for the maintenance of the samples, sterile body fluid should be maintained at room temperature until the culture and other procedures have been performed. And blood culture isolate should be maintained for 30 days, and all the positive culture should be kept for 7 days for further evaluation like molecular typing. And here comes standard operating procedure. This also it plays a very important role in maintaining the quality control. So this is an essential component of the quality control, and it should be written in the format of CLSI guidelines, which is very important. And it should be reviewed and signed annually or biannually by the laboratory director. So based on the SOP, it improves the and maintain the quality of laboratory service to the patients, and it identifies the problems associated with poor work performance, and to help avoid shortcuts being taken when performing tests, and it also provides safety in the laboratory. So standard operating procedure is always important to give the perfect result finally. And each laboratory should have standard operating procedure manuals, and it should include all these points like infrastructure of laboratory, biosafety precautions, disposal of infectious waste, collection, transport, storage of specimens. And in specimen collection, I forgot to tell you, actually there should be a specimen collection manual in every hospital. Even we have this. I forgot to keep that slide actually. And preparation, material rejection of samples, processing of samples, and all these reporting, procedure of quality control, references, these all are included in this standard operating procedure. And finally, quality is everyone's responsibility. And as we know, we have already got the entry level accreditation by NABH. So just a small presentation to, that's why I selected this topic regarding quality control. Hope you all enjoyed, and I thank everyone, our HOD and Dr. Swati Madam, Girisha Madam, and Ratna Madam for encouraging, and I also thankful to all the association people for encouraging, and I have presented this topic. Hope you all enjoyed. Thank you very much. And now the next session is followed by my colleague, Mr. Ramesh Patel, regarding PCR. Thank you.
I have a question for you.
Yeah.
You covered the QC on virology samples, parasitology sample, and mycology samples.
Ma'am, I didn't get you. Yeah, ma'am.
Parasitology and mycology samples.
Equals external quality control we are doing, ma'am. Hello?
QC.
QC we will do positive control, negative control standards. We will run with each test.
Fungal specimens.
Parasitology and
fungal specimens.
Okay. For fungal specimens, we will prepare already the slides, known slides. We will stain it and we will check it out. We will store it.
Okay. For parasite.
Also same.
Okay, fine. Any other questions by audience? It was quite a short and crisp and very informative talk, Asma. Very good. Any other questions by audience? Okay. Fine. There are no questions. Yeah, we move on to the next session. In continuation to today's talk, our one more technician who wanted to tell in detail about the PCR technique. We welcome Ramesh Patel for the same. Let's continue, ma'am.
Can you see the slides? Hello, okay. My slides are visible.
Madam, are the slides visible, ma?
No.
Yes,
Is it visible right now?
Yes.
Okay. Good afternoon, all. Myself is Ramesh, lab technician, ESI Specialty Hospital. I wish all of you that a happy medical laboratory professional week. I'm happy to say that the ESIC MLT Association achieved a part of its goal, that the Junior Medical Laboratory Technologies designation upgraded from laboratory assistant. I'm very thankful to the active members of the ESIC MLT Association. I congratulate each and every laboratory professional to that National Commission for Allied and Health Professional Bill 2020 passed and published in Gazette of India. I'm coming to the today webinar topic, basics of PCR. PCR, its expansion is polymerase chain reaction. PCR is a revolutionary method developed by Dr. K. Mullis, a researcher in California biotech company. The name is CETAS, company name, in the 1983. For this work, Dr. K. Mullis awarded the Nobel Prize in 1993. In 1985, first publication of PCR appeared in Science. PCR is a technique used to generate thousands, million copies of a particular section of DNA from a very small amount of DNA. When we discuss about the PCR, we have to know that DNA and RNA. What is DNA and what is RNA? DNA expands that deoxyribonucleic acid and RNA expands ribonucleic acid. DNA or RNA contains the instance needed for an organism to develop, survive, and reproduce. DNA composed of two polynucleotide chains in two ringed bases, adenine, guanine, and remaining two are pyrimidines, ringed bases, cytosine, thymine. When it comes to RNA, the thymine replaces with uracil. The DNA structure is like this. The two helical structure of DNA backbone is made up of deoxyribose sugar and phosphate. Constituents of PCR reaction: DNA template, primers, DNA polymerase, DNTPs, buffer, cofactors. Steps in PCR. PCR method is based on three simple steps. First, denaturation, second, annealing, and third is extension. PCR cycle is simplified with this temperature cycles. One is DNA denaturation, primer annealing, and primary extension. DNA denaturation. DNA denaturation is a process of separating double-stranded DNA into single strands. When a DNA solution heated enough, around 92 to 94 degrees centigrade, the double strand of DNA unwinds and the hydrogen bonds that hold the two strands together weaken and finally it will break. Process of breaking double-stranded DNA into single strand is known as DNA denaturation. Annealing. When the temperature is lower to enable the DNA primers to attach to the template DNA, primers bind to their complementary sequences. Extension. The PCR step in which 70 degrees centigrade temperature activates are tag polymerase, which extends primers and replicates DNA. The reaction mixture is heated to 72 degrees, which is the ideal working temperature for the tag polymerase. The polymerase adds nucleotides DNTPs complementary to the template. It will happen as like this picture: denaturation, annealing, and extension. Types of PCR. There are many types of PCRs, like around 28 PCRs. Generally, we use in medical diagnosis RTP, real-time PCR, reverse transcriptase PCR, and in this semi-quantitative and quantitative PCR. Applications of PCR: molecular identification, DNA fingerprinting, classification of microorganisms, or the genotyping, prenatal diagnosis, mutation screening, drug discovery, genetic matching, detection of pathogens. When it comes to sequencing, bioinformatics, genomic cloning, home genome project, and when it comes to the genetic engineering, site-directed mutagenesis, gene expression studies. A small video about this basics of PCR.